There are more than 344,000 clinical trials in the United States and in more than 200 other countries. These clinical trials are all different, of course, but they have one goal in common: to be as productive as they can possibly be. Welcome to Podcast 5 of CTO, the Clinical Trial Optimization Podcast. CTO is a twice-monthly podcast that brings together clinical research stakeholders to exchange ideas, share knowledge, and think creatively about how to oversee, manage, and optimize clinical trial planning and execution. Podcast includes discussions with clinical research industry thought leaders and practitioners about how the industry is transforming clinical research design and operations to speed up the delivery of life-changing therapies. I'm your host, Linda Sullivan, and I want to thank you for joining me on this exciting journey to raise the bar on clinical trials and provide an interactive forum for discussing what we do professionally every day of our lives. We hope you'll subscribe to CTO on your favorite podcast platform so you'll automatically get every episode in your feed for free. In this episode, we'll be talking about the design and implementation of pain management clinical trials and how lessons learned can be applied to improve operations and other studies today. So let me introduce them to you. First, let's meet Dr. Nathaniel Katz. Dr. Katz is considered one of the leading experts of treatment and clinical study design in pain clinical trials. He's a neurologist and pain management specialist with a distinguished career at Harvard Medical School, Brigham and Women's Hospital, and Dana-Farber Cancer Institute. He founded Analgesic Solutions with the mission of modernizing the design and conduct of pain clinical trials to advance the scientific quality of pain clinical research and empower effective treatments for patients. He serves as Chief Science Officer of Analgesic Solutions. Welcome to CTO, Dr. Katz.
Thank you very much, Linda. It's a pleasure to be here with you today.
And now I want to bring in Dr. Arturo Morales. Art has 20 years experience working in the biotech pharma industry in all aspects of data science and biology, spanning the entire R&D spectrum. Prior to the WCG acquisition, he oversaw all technical and data science operations at Analgesic Solutions, including the development and commercialization of the company's proprietary quantitative data surveillance system, QDSS, and learning management system. Art started his industry career at Genome Therapeutics as a senior computational biologist. He subsequently led a variety of teams at various companies, including the Novartis Institutes for Biomedical Research, where he was global lead for biology platform informatics and responsible for the integration of all research data into a new data warehouse and the development of new data management and dashboard applications. Art holds a Bachelor of Science in Biochemistry, Biophysics from Rensselaer Polytech Institute, and he completed his PhD in biology with emphasis in bioinformatics and structural biology, biophysics at the Massachusetts Institute of Technology. Welcome to CTO, Dr. Morales.
Thank you, Linda. Nice to be here.
So let's start off, Nat. If you could give us a little bit more information about your background and sort of how did that lead you to decide to start what is now known as WCG Analgesic Solutions?
Sure. I think this story probably begins sitting in the clinic treating patients and being unsatisfied with the treatments that we had for chronic pain, for acute pain, for cancer-related pain. It doesn't really take long sitting in the clinic and trying to handle those terrible problems that you realize that the treatments that we have are not really that good. And I'm sure that clinicians in every therapeutic area can relate to that experience. And so I decided at that point that the best way for me to contribute to the development of new therapeutics would be to start getting into clinical trials, which I started to do and set up a research center. And it didn't take long in that space to realize that clinical trials was more complicated than it seemed from the perspective of the practicing clinician. I initially had this naive idea that if the drug works, well, just give it to the patients and give some others a placebo. And if the drug works, the study will show it. And that turned out to be a very naive notion that unfortunately I think is still widely believed by many people who don't fully appreciate that if your drug works, the trial will not show it unless the trial is designed and conducted in a very particular and mindful way about all the factors that can cause trials to be unreliable. And it was that observation that led me to start Analgesic Solutions and focus on trying to figure this problem out in the pain area, which was my therapeutic area of focus. And then after a decade or so of doing that and discovering a few useful tricks, then to try to make those tricks available to people and companies working in therapeutic areas outside the pain area where really the problems and the solutions are very analogous.
And Art, tell us a little bit more about how your background in biotech and IT kind of brought you to Analgesic Solutions and why you thought it was a good place to come on board.
Sure. So I literally grew up in research. I was doing research when I was in 8th grade at a university, and that was my after school work. And I grew up thinking about scientific problems and really trying to either understand biology or starting to develop therapeutics. And it was always about asking the question, why not do this? What can we do here? Why is this working? Why not? And there was always this big wall between research and development. And I was actually in some companies that we thought that we were going to break that wall down. But after 20 years in research, it was always, we'll do all these questions, we'll do everything. We're trying to do as much as we can because we know clinical trials fail often, or not the fault of the drug, just anything could happen. And when Analgesic Solutions opportunity came up, it was a unique place. It was sort of the perfect storm of being able to see, well, not only is this company thinking that asking questions is correct, but it's the right thing to do during trials, but also we are in an opportunity that we can actually change the way clinical trials are run. This was a few years ago, and it was always this thinking of a clinical trial as a black box, and we were saying, well, what if we open that black box? What if we see what's going on and ask questions and innovate and make it more effective? And as we said now, is give the drug the best chance to show that it works. And thankfully, so far it's been a happy story that we've been able to show some things. I'm excited to join at first, and I'm so excited to be part of it.
That's great. So, Nat, you started the company wanting to figure out how to design trials better. So what were some of the things that you felt were not being developed properly, giving a drug its best chance to demonstrate that it was effective?
Right. Great question. Well, we looked at a lot of different factors. We were really very open-minded about what could be causing the results of clinical trials to be so unreliable to the point where some of them literally failed to show that drugs that are known to work. We explored a variety of different areas without really having any preconceived notions where we might find problems to be. And everything kept us leading us back to the patient, where we had spent a lot of time over the last 50 years or so developing questionnaires and other forms of assessment tools to assess patients' symptom intensity, whether it's intensity of pain or nausea or sleepiness or whatever symptom it is. But somehow the field had forgotten about the patient and lost track of the fact that the questionnaire is not being filled out by the finger of God coming down from heaven. It's being filled out by a very fallible human being. Some humans are much better than others at using these questionnaires that we've created, validated or not, to report how well they're doing. And our earliest important discovery, I think, was that patients, in fact, do differ in terms of how accurately they can report their own symptoms using whatever instrument you give them. And that led us to a series of discoveries. But probably the most important one was that you can train patients to do that better. We've successfully demonstrated that a number of times. And now that's actually become one of the leading services that we offer is training patients to report their own symptoms more accurately, which is not really a surprising thing. And it was sort of hiding in plain sight all along. But in fact, that approach is now informing how we bring patients on board into clinical trials. It's informing how we monitor their performance during the course of their clinical trials with our central statistical surveillance tool. And it's even informing how we've designed our ePro and eCOA systems to make sure that if patients enter information that makes no sense, we can intercept it at the time and teach the patient how to use that opportunity to more accurately convey how they're actually feeling. And that appears to improve the success rate of clinical trials.
So that's really quite interesting. It's not a matter of they're just forgetting to fill the forms out, but in terms of If I say my pain is a 5 today and it's a 7 tomorrow, did my pain really change? Are you saying that the actual data itself was not as reliable as one might think?
Yeah, the striking error that the field of clinical research has made over the last few decades is the belief that because something is subjective, there's no way to know whether it's being reported accurately and reliably. That's that's a fundamental misconception that has kept clinical research methodology in the dark for a long time. Once you discard that misconception and you realize that, if you give a patient two different scales at the same time to report their pain intensity, for example, and on one scale they say their pain is high, and the other scale they say their pain is low, it's obvious that that patient is not reporting their symptoms accurately, even if you don't know exactly what their symptom intensity is. Or, for example, if they say that their worst pain in the past 24 hours is a 3 and their average pain in the past 24 hours is an 8, you don't have to know what that patient's symptom intensity was to know that they're not reporting their symptoms accurately. And then the striking finding is that with just a little bit of training, you can prevent those problems from occurring. And when they do occur, and you pick it up with your central monitoring approaches, with a little bit of remediation effort, most of those problems can be corrected. And then all of a sudden, you get cleaner data just because you've discarded a misconception and you're operating with more clarity.
So Art, maybe from the data side, you can tell us a little bit more about, as you started to look at this data, and you came out of the biopharma side. Were you surprised at what you were seeing?
I was. And one of the things that I always complained about, which is why I always thought that this would be a much harder problem and almost intractable, was that, from pharma, we always want to boil the ocean. Nat and I have always had this great discussions, the fact that, I'm trying to boil the ocean. He's trying to just start with the teacup. There's too many columns and not enough rows from a data perspective on a clinical trial that it's very difficult to find out where the problems are. And the thing that we really, the hone in, the key here was to know, well, where it really is, where is the clinically relevant problem? And when we started looking at that data, it was very apparent. I mean, coming from a non-clinical perspective, just seeing that it's obvious problems that anybody, with just a minor 10-minute explanation, well, of course that makes no sense. But we were missing them because generally the industry was focusing on trying to fix everything at once. And I think that was probably, for me, one of the biggest sort of learnings of this process is that you need to pair mathematical significance with clinical relevance. And that's been really powerful for us.
That's great. So tell us a little bit more about what types of organizations and size of organizations do you work with? You sort of have this offering now. Is it mostly sponsors that you're working with, or academic medical institutions? Like, who actually is utilizing these types of services right now?
I would say that most of our partners and collaborators have been pharmaceutical companies and to some extent medical device companies that actually have to get real clinical trials done. And if the treatment works, the trials have to work. There's no tolerance for failure. Our clients range from the world's largest pharmaceutical companies to, two guys and a cat, sitting in a garage in Cambridge somewhere, and sometimes two cats for the more heavily resourced small startups. The sorts of collaborations differ from the larger companies to the small companies. Everybody, uses our training services right now because that's something we've been doing it for so long and refined it over such extensive periods of time and generated so much data around the patient and staff training that we do that's something that's nobody wants. Very few people want to try to replicate that on their own and take their chances. So that's something that all size sponsors work with from a risk-based monitoring and central monitoring perspective. Companies that have their own teams in place, our role with them is often more consultative or providing our software tools to them. And for small companies that aren't as well resourced, we're often in the trenches with them providing risk-based monitoring and related services. And for everybody, we also support people with clinical trial design services as well. Since over the last quarter century, we've seen so many things go wrong that we've got the battle scars to help people avoid making the mistakes that others have made before them.
Great, thanks. We're going to take a short break right now so I can tell you about the upcoming MCC Clinical Trial Summit. When we come back, we're going to talk about how COVID has impacted what you're seeing going on with clinical trials. If you've enjoyed this discussion so far, please join us at the MCC Clinical Trial Risk and Performance Management Virtual Summit taking place September 8 through 10, 2020. We've taken our successful face-to-face summit format and completely retooled it to deliver a unique, engaging, interactive multimedia experience. Beginning on August 26, you'll have access to pre-recorded case study presentations about quality by design, risk-based quality management, as well as vendor selection and oversight, and risk-based approaches to selecting vendors and sites for audits. Then, on September 8th, you'll move to the 3-day virtual live portion of the summit. FDA keynote speaker Dr. Jean Melinda, Senior Policy Advisor for the Division of Clinical Compliance Evaluation in the Office of Scientific Investigations, CDER, will discuss whether adoption of QBD and RBQM approaches are improving clinical trial quality. Next, you and other participants will gather in virtual breakout rooms to meet with the pre-recorded session speakers and engage in facilitated community discussions about various risk and performance management topics. And some participants will opt to take it to the next level, to roll up their sleeves and join a virtual working group to build a solution during the summit. Participants will prep for the working group by viewing a selection of pre-recorded presentations and gathering information prior to the live session. Topics to be explored in this format include centralized monitoring KRIs and QTLs. What do you measure and what's useful? RBQM conversations in the field. Applying sales training concepts to real-work problems. Reassessing risk during study conduct. When and why? How to define and estimate IT system benefits before selection, and what are the most important questions about vendor performance your organization seeks to answer. Finally, for those of you who participated in the summit in the past, let me reassure you that the data analytics team exercise, aka Follow the Data Trail competition, will be run at the virtual summit. So don't miss this opportunity to participate in this unique event. It's time to meet your peers, collaborate, share ideas, and think. To register for the summit, visit www.mcc-summit.com. I want to make sure you got that. It's www.mcc-summit.com. And here's the kicker. If you subscribe to the CTO podcast, remember it's free, you just have to click on subscribe, you can use the discount code POD15 to receive 15% off your registration fee for the summit. That's right, just subscribe and use the code POD15 and you will receive a 15% discount on the summit registration. So visit www.mcc-summit.com for details today. Hey, welcome back to CTO. So how has the COVID-19 pandemic impacted what's going on with the trials with your customers?
Art, why don't you take that one, since you've been doing a lot of great work with using data science tools to help sponsors figure out what's going on with their trials and the current situation.
Sure. So one of the things that we saw, obviously, operationally, when all the sites closed, trials needed to keep running in some cases, or they could pause depending on the trial. So that gave sponsors an opportunity to really reevaluate, in some cases, how were the trials running. It gave an opportunity to say, could we make any changes now? The regulatory agencies also said things have to change. Patients may not be able to go into the site. Can you still do more telehealth? Can you do more virtual visits or virtual trial-like things that may need to be implemented in a hurry, number one. So we've seen that across the industry. But in the case of looking at data quality from a trial, we started to see sponsors that said, well, now that I needed to stop, I want to restart, but how is my trial going? This is an opportunity to, now that they slowed down the train, well, should we let some passengers out? And that we were able to look in a couple of examples to look at how the performance of data of the site from a data quality perspective, for example, was going to say, well, if you're going to reopen the study, instead of focus on this subset of sites that are doing really well, these other sites may not be worth it. And obviously, you're still looking at this in a blinded study, so you're still trying to understand what's happening. But the idea of being able to have an orthologous view into what's happening in your trial and make decisions to optimize things, because now instead of running 100 trials with travel problems, you may not be able to go visit all 100 sites. Your monitoring plans need to be changed. So can you bring other tools that are more data-based and science-based to really help you sort of prioritize and figure out what you can do and how you can do it? So that's been a big change for us.
Yeah, and what I would add to Art's comment is that everybody always wants to know who their good sites are and who their bad sites are. But then when you ask them what they mean by that, or how they would know if a site was good or bad, things get very vague very quickly, and people understandably step onto the firm ground of operational metrics, which is usually like who's enrolling the most patients, who's got the lowest number of screen fails, who's getting us their data in a timely manner, et cetera. And those are all very important things that people should be focused on, which is good. What's missing, though, is who's going to separate drug from placebo. Well, how would you know that in a blinded data set? Actually, you can know that. And the way that you can know that is that whoever is performing their study assessments, their diagnostic assessments, the actual study procedures that have to be performed accurately and reliably, you can tell, even in blind data, which sites are doing a good job with those activities. Those are the ones that on average will separate drug from placebo, and who's not. And it is true that those characteristics don't always go together. There's some sites that are great operationally, but actually they and their subjects and the performance of the study procedures that matter for your primary endpoint, they don't do a good job. Whereas there are other sites that have only enrolled a small number of patients, and maybe they're a little slower in some ways, but actually their data is crisp and clean from a accuracy and reliability perspective. So sponsors have found it very interesting at this time to echo what Art is saying, that we can actually provide them measurable data on which sites really are good and bad from a true data quality perspective. And sponsors have found that very informative in terms of planning how to emerge from this current situation that we are all in.
Well, one of the things that we've certainly heard in our discussions at MCC is the more use of telemedicine, remote visits, trying to be more flexible in the study design. I imagine that you're seeing an uptake on this too. And what are the things that people should be concerned about as they move towards maybe implementing some ePro things that they haven't had before, et cetera?
Maybe I'll start, Art, and have you follow up.
Sure.
So if you're going to be shifting the modality of patient assessment and the location of patient assessment, then from a psychometric perspective or a measurement perspective, your measurement tools may not function the same way. So just by virtue of the fact that you're assessing the patient in their home environment versus in the clinic environment, you can find that variability changes, measurement of symptom intensity changes, et cetera. And I would foresee that ultimately regulatory agencies will be asking pharmaceutical companies. Right now they're saying, well, go ahead and get your data any way that you can get it. Of course, that's compliant from a regulatory perspective, and people are doing it. But then when the time comes, this question is going to be, well, yeah, I know you listened to us and you went out and you got your data, but how do you actually know that it's quote unquote valid? Which means, how do you know that the measurement performance has not actually shifted while you're getting your data a different way? That is a concern. There are ways of knowing that. There are ways of assessing that, even in blinded data. And I would encourage sponsors to take a defensive position and say, yeah, we will go forth and using technological approaches that Art will probably say more about in a minute. We can get data from patients. However, what are we doing to train the patients and the staff to make sure that the quality remains high? And what analytic approaches or monitoring approaches are we taking to make sure that we're going beyond just missing data, beyond do we have the data or not, to are our measurement approaches performing the way that we expected them to perform, and if not, what are we doing about it? So that's where monitoring the actual, psychometric performance of our scales as we shift in and out of these different modes of data collection is going to become very important. And it's going to be hard to figure out, 2 years down the road when you're submitting the data. So now is the time to make sure you're building in those protections to ensure your data quality is good as we're shifting back and forth. Maybe Art, you wanted to chat a little bit about our technological advances and to help ensure that we actually can get good quality data from patients regardless of their situation.
Sure. I think there's two things I wanted to mention here. One is that really the onus on the focus of the monitoring really obviously needs to switch to patients and the patient's data. The actual performance of the site may not be as important because the patients may not be going to the site. So a lot of the stuff that will be monitored before, because it was available, may not be as relevant. And now you have to guess in many cases, if you don't know better, what is it that you should be monitoring that may have an impact on the data quality. The other part is there is going to be a shift in data quality regardless. The expectation is if you're going from something that was happening in a clinic in one setting and you shift to a different setting, we expect a change. The question is, what is an outlier then? So if you think about defining an outlier based on previous data, so you know what your patients look like before COVID, and now you shift not only to the fact that they're doing it at home, but maybe you introduce a different ePro to collect more data, or you're doing telehealth. That data is going to have a different shape of data quality, or may have a different shape of it. So it is really important to compare your new data compared to it in the context of the entirety of the population of data that's happening at that point. If you compare to the past, maybe everything will look like an outlier. That's not a bad thing necessarily. So really taking into account the fact that your baseline is shifting, it's probably one of the biggest challenges that we're going to see in terms of analyzing data and identifying what is really a problem. Because just because it's changed doesn't mean it's bad, because your entire population may have changed. So now finding those outliers becomes more difficult. We haven't used statistical process control, which takes that into account. There may be other methods. There are other methods out there, but this is something that for me is of vital importance because, again, you're chasing something that is changing at the same time.
You raise a really good point, and we've certainly had conversations at the MCC about how do you use statistical process control when you don't have data yet. It's sort of that catch-22 situation, and I think it'll be interesting to see how organizations handle that situation. Now you have a new norm, but what is it? You don't know what it is until you've done it for a while.
Yeah, so I expect that our ability to detect subtle outliers will decrease temporarily, but then soon enough, as you get more data that's collected, obviously you should come back to your power. But yes, this is a shift that the agency realizes it's a transition period, and we just want to minimize that and maintain as much sensitivity as possible.
And I would add, and this actually echoes a point that Art already made earlier, in that the most useful central monitoring methods result from the marriage of statistical monitoring techniques and clinical science. And that can take a number of different forms. So, for example, if you're doing statistical monitoring, which we do, and you find that, the variability of symptom reporting in some site all of a sudden is different than other sites, well, how do you, there's an interpretation piece that's needed. How do you interpret that? And if you have a clinical science team looking at that data and they find that it's really just an isolated instance, the magnitude is not that high, and they're familiar with the research on what magnitude of these sorts of problems actually end up interfering with your ultimate study results, then they may be able to say, yeah, that's statistically significant, but it's really not clinically significant. So let's just keep an eye on it for now. Or they may be able to triangulate between multiple different signal to say, yeah, there's a problem, but it's really only limited to that one variable. All these other variables actually suggest that the study results overall are healthy. Let's just keep an eye on it. Versus, ooh, that looks really bad. We know that a signal that magnitude can interfere with study outcomes. We really need to jump on that. So we do have to marry statistical science with clinical science to most powerfully ensure that our clinical trial results are accurate and reliable, especially when there's this huge societal perturbation going on now, right in the middle of everything.
Well, that's a great point to kind of think about here. I want to start to wrap up things and just think about whether there's some lessons learned that are happening right now in the COVID experience, and whether you think some of these will have longer term implications in terms of planning and executing trials in the future.
After you, Art.
Sure. So one of the things that I find that we find in a couple of examples already is we've been talking to sponsors about how how to monitor during this checkpoint period, what we call the checkpoint analysis. And quite often some of the questions that come from the sponsor are, well, how could you? This is really interesting. This could be really useful. What can you do? And our answer in many cases is we're limited by what's in the protocol. Obviously, we're not going to introduce any interventions that are subject facing that would impact that. So the earlier these kinds of services are implemented, the better a chance you have to correct issues as they happen. So one of the learnings that I think the industry's probably slowly coming up, and it's been happening, but this I expect will accelerate things, is to really be thinking ahead of the study starting. What can we do to mitigate the risks? Not only do we monitor the risks, how do we mitigate them? And write those things in the protocol, write those things in trainings, and try to stave off as many of those data quality risk as possible through training, through having mitigations in place before they happen, so that you can react quickly. Because now, obviously, this forced all of us to look at trials as they're going, and we've identified things like, what, you have been monitoring this, you could have, and you were able to do something. You could have addressed this 6 months ago, in 10%, when only 10% of your patients were enrolled. You happen to have cut a break because 50% of your patients were enrolled. You still have another 50%, but your hands are somewhat tied unless you're willing to do a big protocol modification, etc. But if you think ahead of time, these things may happen again. So preparing for this is it's not about being a prepper, but it's about being prepared. And that's probably one of the big things that I'm seeing now.
Matt, your thoughts?
I would echo that. I think on a very practical level, if people would just put patient training as a study-related procedure, it doesn't even have to be any more fancy than that. Then you're covered as training evolves. And of course, if you do end up creating new training interventions that are subject-facing, the IRB will need to review those. But in most cases, you will not necessarily need to amend your protocol. That approach gives you a lot of flexibility. And I think the other big lesson that we've all learned is that we have to design our studies in advance and get everything lined up such that if there is an unforeseen interruption of patients' ability to get to clinic, there is no interruption in our ability to collect data. Obviously, there's certain kinds of, you can't do a biopsy in the patient's living room if you're not there, but a lot of the patient assessment data, even based on interviewer reported measures, can be collected remotely. And so just pre-planning that in advance and making sure you've got a good clinical science team, ensuring that your ePro and your eCOA systems are designed in a way that are going to ultimately make sense to the patient. And we do see some funny things that people try to do. Having all that lined up front, it's like the old saying in medicine, an emergency prepared for ceases to be an emergency. And so if you're prepared for these things, then it doesn't feel like an emergency when things happen.
Great. Well, in closing, we're going to do what we call the trio, which is a set of 3 questions that we ask every guest at the end of the podcast. So let's start off with Nat. What do you think is the biggest challenge facing clinical trial industry today? What's the best new innovation? And what's your top optimization takeaway?
Oh, Linda, those are such hard questions, but I'll take a crack at it. From my vantage point, the biggest challenge, since I work in optimizing clinical trial design and conduct, the biggest challenge is that we don't really have a vocabulary for discussing whether our clinical trials are accurate and reliable. the word, reliability of clinical trial results appears 9 times in the ICH E6R2 revision, but there's no definition of that concept anywhere in that document or any other published documents. And so I think that leaves sponsors and CROs very uncertain about how to know whether their ongoing trials are being done accurately and reliably and thereby fulfilling regulatory expectations. So adopting simple approaches to understanding when what you're doing is accurate and reliable and implementing it, I think that's a big challenge, although thankfully there has been good progress. In terms of your second question, which is what is the best new innovation, I'm going to be very kind of selfish and biased here, but I think a really important, maybe it's not the best, but a really important innovation is remembering that the patient is really the sun of our clinical trial solar system. And everything really has to revolve around that patient, including remembering what we're asking those patients to do and making sure that we are tracking and providing support to make sure that they can do what we're asking them to do in a fashion that's adequate enough for the study results to be successful if the treatment works. So patient training and patient monitoring, I think those are a family of exciting new innovations. And for your third question, which is what is my top optimization takeaway, I guess in the interest of conciseness, I'll just repeat my answer to question 2, and that refocusing on the patient quantitatively.
Great, thanks. And Art?
I agree with everything that Nat said. I think I would add that the biggest challenge that I see is really coming to terms with the fact that the definition of what a site is changing. And if we do not agree that it's changing, we're just going to do the same thing over and over. And when the next pandemic happens or the next disruption to our supply chains happens, we're going to be in the same boat. So what is a site? It's a site of place. It's a site, a combination of being at home versus being at a lab, et cetera. That is the challenge for us to define and really to adjust our processes, our distributions, and our protocols, et cetera, to deal with the fact that a site may not be at a location anymore. In terms of the innovation, I think the monitor telehealth is what really is the key. We all quickly switch to telehealth. Okay, yes, we can have a one-on-one conversation and see our patients on the other side of the screen. However, without additional monitoring, you're actually losing from what you had at the site. But with continuous monitoring for data quality and things that matter, like what we're trying to do and what the MCC is trying to do, what are the right metrics to monitor? I think that's going to be the innovation here. That's how we are going to be able to maintain the quality of the trials and protect the outcomes as we go from wherever they were happening before at a site to wherever they will be happening now or in the future. And I think and on the third question, it's the same thing. Is how do we get there? Is again by being smart about where is it and marrying the data science and clinical science together, because one by itself is not enough. The clinical science will never be able to work with all the data that's coming through. You can't analyze everything. So you have to somehow filter that first. And the data science is going to help do that. But then obviously, how do you filter? You need the clinical science in there.
Well, great. Thank you very much. I want to thank both of you for a very interesting conversation. I'm sure others will be contacting you with some follow-up questions, and I think you both will also be at the MCC Summit, so people will have an opportunity to ask you questions there. So once again, thank you for joining me today. I'd also like to thank our producer, Michael Levin Epstein. And finally, we want to hear from you. Rate us on iTunes. Are there topics we should be discussing? Guests we absolutely have to have on the show? Let us know by emailing us at lsullivan@metricschampion.org. This is Linda Sullivan, and we'll see you next time on CTO, the Clinical Trials Optimization Podcast.